Dear delegates, good evening. A warm welcome to this afternoon session, which is about aquaculture. We have 3 presentations. So let's invite the first speaker, who is Apostolidi, Miss Apostolidi. Is a study entitled Development of qPCR Assay for the Detection and Quantification of Tenacibaculum maritimum.
Good afternoon. My name is Apostolidi Evangelia, and I'm here to present you the development of a qPCR assay for the detection and quantification of Tenacibaculum maritimum. I come from the Veterinary Research Institute of Thessaloniki. Regarding the pathogen, is a gram-negative filamentous bacteria of the Flavobacteriacea family. It affects a large range of species with commercial value, such as sea bass and sea bream. As you can see in the photos, it produces distinctive gross lesions on the external body of fish, such as mouth erosion, frayed fins, ulcers, skin necrosis, and tail rots. It results in substantial losses across the global aquaculture sector, including, of course, the Mediterranean area. The diagnosis is based on culture-dependent techniques. However, the isolation is not always successful, and definite diagnosis requires colony isolation on specific media, such as Flexibacter maritimum medium, FMM, or media supplemented with antibiotics. There are available PCR tests, however, they have limited or not at all capacity in quantifying the pathogenic bacterial load in infected fish or in environmental samples. Therefore, the aim of the present study was the development of a novel, rapid molecular method using real-time PCR with the use of a Tachman probe for the specific detection as well as quantification of Tenacibaculum maritimum. For this purpose, we used the gene bank databases and online available softwares in order to choose the most specific gene area. We were between 6 other areas, but finally we selected the translation initiation factor IF2 of INF-B gene. Therefore, we designed a primer pair along with a Tachman probe, and the method amplifies part of that region that is 161 base pair long. The reaction was performed in a final volume of 25 microliters containing kappa probe fast qPCR mix, 300 nanomi of each primer and probe, and approximately 20 nanograms of bacterial DNA. The thermocycling conditions were optimized, and you can see them in the slide, and the analysis was performed using CFX Manager software. As controls, we used non-template controls in duplicate in each run, and negative control samples that were originating from fish pathogenic and non-pathogenic bacterial strains that were previously identified by DNA sequence. This regards the species Vibrio, Staphylococcus, Photobacterium damsellae, Escherichia coli, Pseudomonas, and Lactococcus garviae. As positive control, we obtained a DSM reference strain. We started with the cultivation of the reference strain in selective media FMM, and then we quantified it using the standard plate count method. The quantified bacterial culture was then used for the extraction of quantified bacterial DNA. The final concentration was approximately 10 to the power of 7 copies per microliter. In order to create the standard curve for the quantification and to estimate, therefore, the amplification efficiency, the dynamic range of quantification, and, of course, the detection limit of the method, we used the serial 10-fold dilutions of the aforementioned quantified DNA extract. The standard curve was performed at least 3 times. The PCR products of the method were also sequenced by an ABI genetic analyzer for the evaluation of the method's specificity. As you can see, the method showed a very high sensitivity of 1 gene copy. That means 1 bacterial cell of Tenochibaculum maritimum. The method showed a linear range of quantification over a 6 log 10 range and a high amplification efficiency of 98.3%. The specificity was confirmed by the absence of amplification products, either on non-template controls or in the negative control samples, we said in the material and methods. The PCR products that were sequenced showed a very high homology, and the sequence has been already submitted to GenBank. So why is this method useful? Recent studies show that tenacibaculosis is one of the most important bacterial diseases along with vibriosis, especially for European sea bass, leading to severe mortalities. In addition, since the common treatment strategy is based upon the use of antibiotics, that can lead to potential presence of antibiotic residues, antibiotic resistant, and environmental pollution. A tool of early diagnosis can help the early treatment of the disease and therefore have a positive economic impact for the fish farm industries. Moreover, nowadays with the climate change, heightens the demand for the development of early diagnostic tools and management strategies. In conclusion, the advantages of the developed method when compared to traditional microbiological methods is its speed and reliability. When compared to the gel-based PCR assays, the higher sensitivity and specificity, and when compared to other available real-time PCRs, it provides the benefit of quantification of the pathogenic bacterial load, either in infected fish or in environmental samples. The gene area we targeted of INF-B gene provides superior sensitivity of 1 copy per microliter, whereas the other 2 available real-time PCR methods that targeted the 16S rRNA gene of the pathogen provide a sensitivity of 4.8 copies per microliter and 2.22 copies per microliter. Its future perspective is its application of qPCR in environmental DNA samples. And I could say we have already started doing that with really good results. I would like to thank you for your attendance. And I have to say, this was funded by the within the framework of the program Prognosis, Correlation of Abiotic and Biotic Factors for the development of a disease prognosis model in fish farming units, funded by the Fisheries and Maritime Operational Program. Thank you again.
Thank you very much for this very interesting presentation. I would like to hear some questions from the audience, please.
Please don't be shy.
Oh, yeah.
Make a question, please.
No, okay. Well, I was wondering, what is the cost for each sample in order to test the cost, economic cost, in order to test a sample?
In order to test what is the cost?
Cost in order to test a sample.
Yes, it's the DNA extraction. Of course, it's around. It depends on what scale you do that.
What per sample?
Yes, per sample, it's around, let's say, €6 the DNA extra, 12 euros, I would say, 10 to 12 euros.
Okay.
It depends if you are research, if you do it on research or it's the common practice of the laboratory.
Let's say that you have a laboratory and you do diagnostic services.
Yes.
So what would be the cost?
I think around between, I would say a range of 8 to 12.
Okay, this is a very good price. I think so. I thought that it would be higher.
Also, its application on the environmental DNA samples. That
Yeah.
means from the water around the farm.
Yes.
The fish farms. So
Okay.
you can predict, since you have
Yes,
such
this
high
is
sensitivity,
very important.
you can predict.
Okay, for future application, this is very important. And concerning the eDNA you just mentioned, do you know how many samples do you need for the eDNA in order to have a reliable result?
We tested some areas.
Okay.
We had at least a triplicate in the start of the farm, of the fish farm, in the end, and also you always need a control point.
Yes, of course. Okay, so okay, with me, any more questions from the audience? No. So thank you very much.
You are welcome. Thank you.